Bueno, buenas tardes a todos. Les damos
Good afternoon. Be welcome on behalf of Cegyr to the third Tribune in the online version. Today, more specifically in our second exchange meeting, the session will be very interesting, very important, with another center which is part of Eugene, Boston IVF, and we will encounter a very interesting program, very topical for all of us, with first level professionals at international level, very well known. So let us you can all make advantage of today's lectures. We thank you very much, Dr. Denis Sakas, for your presence here, for sharing your knowledge. We will review our way of methodology, modality. You can select the language of your choice. You find an icon. You can select Spanish or English. And throughout the webinar, you can send questions, comments, whatever you want to comment in the question and answer icon, Q&A. Please do not use the chat. So now, my co-coordinator, Dr. Andrea Cosia, will take the floor. She is a specialist in gynecological reproductive endocrinology, reproductive medicine, our director of the fellowship program, and she coordinates the oncofertility program of Cegyr, and she works with us. Andrea, thank you. Thank you, Susana, for your kind words. Be welcome. It is a great honor for me to present our speakers. We will start with Dr. Denis Sakas. Let me make a brief introduction. He works in Melbourne, Australia, and has a PhD in philosophy in Melbourne, Australia. He is editor of Reproduction Magazine, scientific director of Boston IVF, and professor of the Department of Obstetrics, Gynecology, and Reproductive Sciences on the School of Medicine at Yale. His presentation is on techniques, non-invasive techniques. It is a great honor.
Thank you, Cegyr, for the invitation. Let me just share my screen. Oh, you've seen a lot of my talk. So it's a great pleasure to be invited to speak to the group, and good afternoon to everybody. I hope you have an Australian translation, because so it might be Australian, English and Spanish today, and actually the speaker after me has an Irish accent, so you will have a lot of English accents today. So today I'm going to talk about non-invasive technologies for embryo selection and possibly the end of biopsy. So this is something I've been interested in for a long time, and I'm going to give the pros and cons today of some of the technologies that are available, and you know, some of the problems maybe of the current way we look at embryos. Just some conflicts of interest. I'm on the scientific advisory board for iGenomics. I'm an advisor to some companies, Legacy, A-Life and Ohana, which may not influence the talk today. I do speak for EMD Serono, and I'm also on the editorial board of deputy editor for Human Reproduction. So we're all familiar with these statistics, and I chose 2015 because it was pre-PGT, let's say, or the big influx of genetic testing in America. But these data don't change much from year to year. So as you know, we're very good at getting cycles started and getting to retrievals, and actually quite good at getting embryos and getting to transfers. But the biggest challenge in IVF is still, I would argue, this period here when we go from the transfer to the pregnancy. And this is a challenge because of many reasons. One, unfortunately, many patients don't have an embryo that will go to pregnancy. But secondly, the embryo that we choose is actually sometimes the wrong embryo. And it may take patients many, many cycles to actually get to that right embryo. So our knowledge of what the best embryo looks at has the greatest impact on success. This period is actually probably the hardest for us to actually judge. So for many, many years, a lot of very smart people have been doing a lot of research into trying to understand which is the best embryo. And there are many techniques that we've been trying to develop and to try to pick the best embryo. And of course, this is the one that everyone I think is the favorite so far of everyone. It's an invasive technique. It works very well, although there are some arguments that it doesn't work very well, but it builds on the thesis that if you can find a euploid embryo, a chromosomally normal embryo, and put it back to a patient, then that is the best embryo. But it's very invasive. If you show this picture to a patient, they probably would be very scared, I think, of what we do to their embryos. So it's a technique that we feel very comfortable doing, but whatever we say, it's still an invasive technique. So what about PGTA? Well, this is probably another talk, but I'll just briefly go about some of the data of actually invasive pre-implantation genetic testing. And there's been many, many studies. There's been randomized controlled trials, matched cohort studies, a statistical technique called propensity score matching, all trying to understand how efficient this technique is. And this is a paper we published several years ago now, just looking at clinically the question of to test or not to test, and how you can counsel patients about the use of PGTA. And basically we took patients coming in for their first cycle, and we knew which patients wanted to go towards PGTA and which patients did not want to go to PGTA. And what we looked at, an analysis of actually, it was a matched control study, and it was like an intention to treat analysis of what happened to these 300 cases in each group doing their first cycle or their first retrieval cycle. Well, not surprisingly, this is what we saw. Well, some surprises, sorry. So if you looked at the under 38 patients and you looked at actually the cumulative live births, so the number of live births per retrieval, what was interesting was actually patients who actually chose not to do PGT actually had a better amount of live births. They had a higher live birth rate. If you looked at live births per transfer, for the young patients, it was not different. For older patients, the cumulative live birth rate per retrieval was the same, whereas for older patients, there was obvious benefits to doing PGT per transfer. And then overall, it evened out. There was still a benefit cumulatively for not doing PGT overall per retrieval, but per transfer, you're obviously getting more value for your embryos. So this sort of brings to light some of the questions we now are asking more frequently about PGT. So this provided some sort of counselling for patients to know when they should do PGT and when they should not. And basically, what is interesting is that I think patients who are older than 38 probably benefit the most. And this is not something new, I'm telling you. Hopefully we've understood this quite frequently. And probably one of the best studies, which was not devised as an intention to treat, because it was actually very focused on actually patients who already had a blastocyst for transfer, was the STAR trial. And this was a very large multi-center randomized clinical trial. We were part of this trial. And not surprisingly, it came to a very similar conclusion to what I just showed before. But this was a multi-center trial. It was 662 women. They actually had to have 2 blastocysts to actually be randomized. So already you were taking back the intention to treat. These are patients that already had samples to biopsy. And the primary outcome was not live birth, it was ongoing pregnancy rate. But what did they find? Very similar to what I mentioned before. Less than 35 years, patients were more likely to actually have an ongoing pregnancy rate, or there was really no difference in if they did PGTA or did not do PGTA. There was a slight favour to patients who did PGT-A per transfer again, because these were patients who had blastocysts. And then finally, when you looked at the data overall, there really was not that much difference between whether you chose to do PGT-A or not to do PGT-A. But still, even though this trial was, 5-6 years ago, the numbers of PGT cases in America and around the world has increased dramatically. And we know that there's a benefit for women over 35 years and older, but it's not that great when we look at some of the data. And of course, there are other ways of statistically looking at this question. And one of these is a pseudo trial, if you want, using a statistical technique called propensity score matching. And again, if you look at propensity score matching and compare PGT-A versus non-PGT-A patients, and this is again looking at different outcomes and looking at patients with live birth, you find that it's favoured PGT-A per transfer if they're older, but actually in younger patients there's no favouring. So the overall story for PGT-A looks like it does favour older patients, but definitely younger patients are probably maybe even losing out doing PGT. Well, there was an analysis done of the STAR trial, a mathematical analysis, and trying to reinterpret the data, and they came up with an interesting appraisal of the STAR trial data. And basically what they said, that according to the model that they had developed, the STAR trial actually demonstrated that transferring only blastocysts classified as euploid after PGT-A actually led to a reduction from 82% to 50% of the live birth rate for competent embryos. So they proposed that in some way doing PGT-A led to embryo wastage. And to be honest, if we looked at our clinical live birth rates per retrieval in the first study I showed, it indicates that the live birth rates were much higher cumulatively in the control patients versus PGT. So there is an indication that doing this diagnosis of PGT leads to embryo wastage. Well, there's also this, what we say in English, I don't know if it translates into Spanish or Argentinian, maybe it's a different animal, I'm not sure, but we say there's an elephant in the room. So everyone wants to do PGT, but there's some fear about PGT that maybe we're not doing something right. And I think now it's coming more and more frequently where the elephant is lying, let's say. So one of the questions that has come out about this sort of amazing technique of next generation sequencing is PGT did not become black and white. It was not euploid and aneuploid. Somewhere in between were these embryos that were a possible mix and what we now call mosaic. So this elephant in the room has actually caused a lot of dilemma for many of us, I think, and especially the clinicians. But there was a really interesting paper published recently. It's actually in American Human Genetics paper, and it was published by the actual genomics group by Anthony Catalbo, and it was a very brave, let's say, trial, randomized trial. And what they did was actually they took all embryos were reported as euploid if they had a chromosomal copy number value below the threshold of 50% abnormal cells. So what they normally would call euploid were embryos less than 20. They would call low degree mosaicism any embryo between 20 and 30% mosaic, and a putative moderate mosaic of 30 to 50%. Now, arguably, in a normal situation doing PGT, many of these embryos would have been discarded, but they decided anything under 50%, we're going to call it euploid. Well, they randomized these patients. They were all single embryo transfers, and many of these patients received known mosaic embryos. And amazingly, well, maybe not amazingly, not a surprise to many of us, the live birth rates were equivalent across the board, indicating that somehow the mosaic embryos are normal or are able to give us live births. And so far, the data that we understand is no mosaicism in actually the fetuses that are being born. So this is, like I said, a very brave and a very interesting study. So where does it take us? Well, it tells us that is biology smarter than our analytical techniques? And again, that's a very separate lecture about how the embryo is actually doing this. How is it tricking us? Or is it that the actual techniques that we're using are just not strong enough, and they're still not robust enough to actually tell us if an embryo is euploid or aneuploid? So something's wrong with PGTA the way we're currently doing it. So there's a dilemma with invasive PGTA. Obviously, many clinics are still doing it on everybody. People have started thinking about, maybe I should do it on select patients. Other people are saying, oh, look, I'm not going to do it at all. So there's a different mentality. And then finally, people are starting to think, maybe we should be doing this differently. And one of the dilemmas with PGTA is, do we do it differently? Do we do it non-invasively? Do we do it completely different and go away from the biopsy technique, which is literally a biopsy of the whole, of a small part of the whole embryo? So one of the arguments that we're having is actually to start doing it non-invasively, but actually don't take PGT as a diagnosis, but as a screening tool, as an analytical tool, and actually start ranking the embryos. So have those of the highest potential that you would transfer first, and maybe you would still transfer ones that you know have a low potential. So what are the theories that we can do for embryo selection that we could actually take away from what we're currently doing? Obviously, we've been using morphology for a long time, so morphology still may have some information for us, and we know that we still believe, I think, that a viable embryo has a different morphology or a growth rate than a non-viable embryo. Obviously, we know that it's highly likely to be one that is euploid, and that's why we do PGT. Some of the other things that we know is that a viable embryo has a different metabolism than a non-viable embryo. And then finally, we know it also has a different uptake or production profile than a non-viable embryo. And of course, many people are still trying to understand the embryo, and I think there'll be other theories that people will find, I think, in the future. So how do we go from this to this? How do we measure something non-invasively? So what are the non-invasive embryo assessment methods that we have available to us now? And we have quite a lot. So this is just a summary of some of the different techniques that we now can use, and we've been using for many years, to actually look at the embryo non-invasively. We can obviously look at its uptake, so we can look at factors that the embryo is taking up. We can obviously look at factors that the embryo is producing. We can do semi-non-invasive techniques where we don't actually take part of the embryo, but we actually look at it in a different way using different technologies. And obviously we can do morphology and cleavage, which we've been doing since day one of embryology. Since Bob Edwards first and Steptoe had the first baby, they looked at the morphology of embryos, but we've actually developed technologies now that are much better at it. So there are also other platforms such as spectroscopy that are proving very interesting in actually looking at the embryo. So we can do two things. We can actually look at the embryo in a different way, so we actually visualize the embryo, or we can look at its surroundings, because we're actually lucky because we grow the embryo in small media and we have very good access to that media. So what about some of the techniques? Well, the trendy, or the new fashion is artificial intelligence. And artificial intelligence is now everywhere. If you pick up your phone and it looks at you, or you look at your phone, it's artificial intelligence that tells that phone that this is you. It's the right person that has the phone. So maybe we can do the same thing with embryos. And there are many people now putting a lot of effort into artificial intelligence-based models for predicting embryo viability. And what's really interesting and quite exciting is that they're doing this just off a picture of an embryo. So this is an example of a paper that was published by Tex Vermiglia using an imaging of an embryo, just looking at the embryo and with very little other information. This is a paper we just recently published doing a similar technology where we just looked at the blastocyst pictures and ranked embryos according to the artificial intelligence predicting tool that said one embryo actually has some hidden features in it that are recognized by artificial intelligence that tells me that that's more likely to lead to a pregnancy or not. And what's amazing is, you know, you look at these pictures, and if an embryologist looks at these, really many of these embryos would be the best embryos. You would think there's no way these embryos would not implant. But what artificial intelligence can do now is somehow recognize features in these embryos that will tell you that one actually has a higher score, a higher chance of getting pregnant than one other one, even though they look to the eye, to me, or other embryologists, very similar. There's also other ways of looking at artificial intelligence, and it's using things like the embryoscope or video imaging, time-lapse imaging. So it marries the image with also the data of its growth pattern. And this is a paper now several years old now, actually, published by David Tran and David Gardner, looking at embryo images and embryoscope data from many positive and negative pregnancies. And what they did, they actually created an algorithm which had features that were linked to those embryos that became positive, and they created an embryo prediction tool, a test set and a validation set, and actually did a ROC curve, an analysis to say, this is what we can predict. And you know, this is incredible if it holds out in clinical trials, but their ROC curve is basically near 1. So they virtually nearly, you know, 93% of the time, they were predicting which embryo would lead to a fetal heartbeat, which is amazing. This technology is now currently under trial. There are many groups working on artificial intelligence using all the data available in an embryoscope or some of the other time-lapse systems, but also people getting these numbers, you know, of high ROC curves with just static images and prediction tools. So I think artificial intelligence has a lot of power in terms of what it may give us as a non-invasive tool. But there are other systems now that have developed in the past few years that are just as maybe efficient, and maybe in the end will be used in addition to artificial intelligence. And one of these that's quite exciting, but again, it's very surprising, is something that was being produced in the culture media that we were unexpected. And that's DNA being kicked out into the media. So doing PGT, but not on the embryo, but on the actual culture media. This was first published by Sunny Z and colleagues at Harvard University. It's now 5 years ago, nearly 6 years ago that this was published. And it was something that was a complete surprise, and many people thought there's no way this is true. But slowly, many groups actually published data showing, yes, there is DNA in the culture media, and that DNA may actually be informative because it matches some of the biopsy results we're getting. So slowly people, you know, worked on this and perfected the technique. There were certain tricks that they had to use. And this is probably the largest study, again, by Carlos Simon and colleagues, Carmen Rubio. It's an interim study of 1,300 human blastocysts. They're just about to publish the final study of nearly 3,000 blastocysts. But quite interestingly, what they did was actually do the biopsy and then at the same time take the culture media. What's interesting here, you can see only day 6 embryos were biopsied, because the way that they had prepared and what people have found is that the culture media and the DNA in the culture media is more informative if you actually culture embryos from day 4 to day 6 or day 7. It doesn't work if you go from day 1 right through to day 6 or 7. So here embryos were always frozen on day 6, and the study looked at the concordance when the biopsy agreed with the culture media, and there were many embryos in this study. So this is what the study looked at. When the embryo biopsy was euploid or aneuploid, did it agree with the culture media? When the embryo biopsy didn't agree with the culture media, or when the embryo biopsy said one thing and again didn't agree with the culture media. So the spent blastocyst media and the trophectoderm had to agree for having concordance. And what's interesting, over the 8 centres that were in this large trial, the concordance rate was about 80%. So 80% of the time, the culture media and the biopsy agreed. And this is interesting, and it's something now that we have to look at clinically. We know that the gold standard is not the embryo biopsy, because we know that if you take a biopsy from the same embryo, the concordance will not be 100% all the time. It could be anywhere between 90 and 100%, and some studies even show lower concordance. So it's very interesting that, you know, people expect 100%, but the gold standard is not actually PGTA the way we're doing it. So this is an interesting technique. We're still waiting for many of the clinical results. Some of the initial clinical results are favourable, showing that it's just as powerful as the PGTA technique. What about other platforms? So getting away from the analysis of genetic material. Well, one of the things that has been of interest for many, many years is actually the uptake and the metabolism of embryos. And we've known that this theory that the embryo has a different metabolic profile, a viable embryo has a different metabolism than a non-viable embryo, has been a very valid theory for many years. And we all know that the production and uptake profile of a non-viable embryo is different from a viable embryo. So what technologies can we use to look at this type of information? Well, this is a study done by David Gardner and colleagues nearly 10 years ago, but actually the first study was done by Yves Menizot and Jean-Paul Renard in 1980, showing that bovine blastocysts that had a high glucose uptake were much more viable than bovine blastocysts that had a low glucose uptake. But you can see here, and this has been shown in many animal models, that if an embryo has a high glucose uptake, it's much more likely to lead to a pregnancy and a positive fetal cardiac activity than one that has a low uptake. So metabolism is a key marker of embryo viability. The trick is, how do you measure that? These technologies are very difficult to do that we used in this study, and there's probably only a handful of labs in the world that can actually do this technique. So if you can get that information in another way, maybe it would actually be beneficial. Well, we've been working on a technology that actually can probably get that information. It's called fluorescence lifetime imaging microscopy. It's a new type of microscopy. And to cut a long story short, it's a candidate technology that measures the mitochondrial activity of cells, in cells. And in particular, it measures NADH and FAD. So we can do a metabolic screening of an embryo non-invasively, because one of the things that happens with NADH and FAD is actually they're autofluorescent. So we don't need to add anything to be a marker for the embryo. We can actually use its autofluorescence properties of NADH and FAD to actually measure the metabolism of embryos. And the way we do that is using this type of microscopy. And I'll show some of the data that we've got over the past few years. So what do we measure? So normally when you measure fluorescence, you're really measuring how bright something might be. It's very bright, it's not as bright, and you can judge that. But fluorescence lifetime imaging microscopy does something different. It measures the brightness, but it also allows the information of capturing when a photon's in an excited state and how quickly it actually takes to decline from that state. So it actually is capturing much more information than just the brightness. It's able to capture the fraction bound of NADH or FAD to the enzyme itself within the mitochondria, and it can measure if NADH or FAD actually have a short or long fluorescent lifetime with the photons. It also actually, because we can divide NADH intensity by FAD, we can also measure the redox sensitivity of an embryo. So we get a very holistic picture of the way the mitochondria are functioning in an embryo. This is just an example of an image, but what we do is actually we can look at embryos, and these are mouse embryos cultured under this microscope, and you can see NADH on the left and FAD on the right. And one of the first things you see very early on, and I'll just show this again, is the NADH is much brighter in the earlier stages compared to FAD. And then you can see, hopefully, that there's a switch in the metabolism. And this is actually well known within embryos, that embryos metabolize in this fashion, that they actually, around the compacted stage, they switch their metabolic profile. But we can actually use this technology to look at human embryos. And we don't look at human embryos all the time. What we do is we actually, at the blastocyst stage, we can take a quick scan of the blastocyst. And this is just an example. So we actually take a scan of the blastocyst. We can actually get information of how the trophectoderm is metabolizing, how the inner cell mass is metabolizing, and we can create a metabolic picture of a blastocyst when it's being tested. So we have a lot of data now on human blastocysts, on discarded human blastocysts. And one of the things we've just looked at, just published 2 recent papers on just looking at the metabolic state of human blastocysts measured by this microscopy. And one of our fellows, Jamin Shah, has just published a paper where we looked at comparing euploid and aneuploid human blastocysts. And I'll just show you briefly some of the data we have from that. We're also actually conducting a large trial on this technology where we're actually measuring the metabolism and transferring embryos to see if certain metabolism was linked to live birth rate. But the data I'm not going to show today. One of the most intriguing things that you can already get is we can actually look at the metabolism of the inner cell mass versus the trophectoderm. And we can look at certain parameters that we measure with the FLIM microscopy, and these are all in the same embryo. You can compare the inner cell mass and the trophectoderm, and you can see here the blue inner cell mass has a very different metabolism compared to the trophectoderm. So already at the blastocyst stage, we can see differences in metabolic activity between the inner cell mass and trophectoderm. We also see differences between day 5, day 6 embryos. We see differences between expansion rates, and we see a lot of differences between embryos from the same patient. So this technology is proving quite powerful in giving us more information about the metabolic status of a human blastocyst. And we've learned actually a lot about how embryos metabolize and human embryos metabolize. This is some, obviously there's not many euploid embryos here, but we have this data, and we have some other data that we've published and we're investigating now. But we do have some indications that some of the parameters we measure show differences between aneuploid and euploid human blastocysts. These particular parameters were not significantly different, but we have a number of parameters, including the redox ratio, and some parameters looking at the NADH activity and FAD, that are indicating that a euploid embryo has a different metabolic activity than an aneuploid embryo. So all this put together, it gives us some light on how the human blastocyst metabolizes and how it may actually be, this information could be used to pick which embryo is viable versus one that is not viable. This is not the only fluorescence technique. This is another technology which actually is broader than the one we use. It's called hyperspectral microscopy. Again, it looks at autofluorescent proteins, markers from cells, but it can look at more of them. And this is a proof of principle analysis on bovine embryos that were cultured in different environments. But this same group have actually recently published a paper looking at this technology and showing that in mouse embryos that are actually euploid and aneuploid, they can see clear differences in the euploid and aneuploid mouse embryos. So finally, this is not the only technologies, but this is a really nice paper by a group in South America using the culture media and mass spectrometry, using that mass spectrometry to look at the culture media of embryos that were euploid and aneuploid, showing that there are factors in the culture media that mass spectrometry can differentiate between euploid and aneuploid embryos. What they did, they had 149 culture media from euploid and 165 culture media from aneuploid embryos. These were analysed obviously with PGTA, but they looked at the secretome or the culture media using mass spectrometry. They were able to pick 12 candidate markers that were able to tell them more clearly what euploid and aneuploid embryos were like in their culture media. And then they used these markers to create an algorithm, and they could use these spectral patterns. And the data analysis revealed a high degree of complementary regions between the euploid and aneuploid embryos. And what was really interesting is that they were actually able to distinguish with quite a good degree of sensitivity the positive euploid and the negative aneuploid embryo. So even showing culture media, this is, you know, a very heartening technique. So there are a lot of techniques now there that will allow us to look at actually non-invasively embryo quality and euploidy or aneuploidy from embryo and their viability. So in the future, how are we going to rank these 4 embryos from patient A? I think we're going to use a non-invasive ranking system using some sort of viability scoring system. And these embryos will actually be ranked as the highest potential for live birth, whether it's by artificial intelligence, metabolism, their culture media, or a combination of those factors. And we will know which embryo has the highest potential for live birth and the lowest potential for live birth in each embryo. So in the future, how will we actually transfer the best embryo? I think the best embryo will be ranked as to its highest potential, and I believe that we'll actually do this non-invasively in the future, and the biopsy technique will be a thing of the past, hopefully. So thank you. These are, you know, obviously a lot of collaborators I work with, and I'm lucky to collaborate with for over many years. In particular, the team from Harvard and Tim Sanchez for the work on FLIM microscopy, Carlos Simon, Carmen Rubio from iGenomics, who we do a lot of studies with, and a long-time collaborator, David Gardner, who we do a lot of work with on metabolism. So thank you. And if you have any questions, if I don't get to answer them today, please feel free to contact me on this email address. So thank you very much for the invitation again.
Excellent. Thank you very much for your presentation. It's been spectacular and very interesting.